Hello and welcome to Mr. C's Semester 2 biology class. We started with the cell membrane. The cell membrane is a phospholipid bilayer, which means two layers of phospholipid which have phosphate heads and fatty acid tails. It also adheres to the fluid mosaic model, which means all of the elements slide against each other, and the mosaic means that there's lots of different types of molecules like cholesterol and proteins, and glycoproteins and lipoproteins. Cholesterol is for stability and has proteins for transport and communication. The cell membrane is also semi-permeable that allows for some molecules to pass through but not others. The shape of the cell is also important because of the rule the higher the surface area to volume ratio, the increased rate of interaction. And in this case, we're going to be talking about movement in and out of a cell. So when we look at movement through membranes, the very simple one is diffusion, which is any molecule moving from a high concentration to a low concentration along the concentration gradient, and it's passive, which means it requires no ATP. In our course, we discussed osmosis, which is the diffusion of water. Water moves from a high concentration of water, which we know as a hypotonic solution, to a lower concentration of water, which we call a hypertonic solution, along the osmotic gradient through a semi-permeable membrane, and again it's passive, which means it does not require ATP. And here we have a little diagram on the right-hand side showing what happens when you put a red blood cell into a hypertonic solution. It shrivels up in a process we call crenation. When it's in a hypotonic solution, it will swell and may burst in a process we call lysis, and in an isotonic solution, it should stay the same. Then we moved on to enzymes. Enzymes are organic catalysts. Organic means it's a molecule with a complex carbon backbone, and the word catalyst means it speeds up the rate of reaction. It speeds up the rate of reaction by lowering the activation energy, which is the energy needed to break the bonds in the substrate so they can be reconfigured into products. The reactions can be catabolic, which is the breakdown of larger molecules into smaller ones, which is also exergonic, that means to release energy. Or it could be anabolic, which means to build up smaller molecules into larger ones, which is endergonic because it absorbs energy. We spoke about the lock and key theory, where the substrate enters the active site. It forms the enzyme substrate complex, which leads to the release of product. The active site is unchanged and is therefore reusable. The substrate fits perfectly into the active site, and we call this specificity. Some enzymes are reversible if the products build up too high. And here's a diagram of the lock and key theory. Then we moved on to some enzyme processes, and first we came across photosynthesis. Photosynthesis turns light energy into chemical energy. Producers produce glucose, and autotrophs use their glucose for cellular respiration. Photosynthesis occurs in the chloroplast. It has stacks of thylakoids called granum, plural is grana, and it also has a fluid matrix called the stroma. Photosynthesis has two separate stages. It has the light-dependent reaction, which requires light and occurs in the granum. Chlorophyll absorbs the light, which ejects an electron, and that electron makes water split in a process called photolysis. This makes hydrogen ions and oxygen molecules. It also releases 18 ATP. Then comes the light-independent reaction that occurs in the stroma. Carbon dioxide is taken from the atmosphere through the stomata and is then reacted with the hydrogen from the light-dependent reaction. It also uses the 18 ATP made in the first reaction. This makes glucose and water. Here we also have a great picture of the cross-section of a leaf, and you should know this. Then we looked at the movement of water through plants. Water moves from the roots to the leaves in an upward direction only. Water travels through the xylem vessels in a passive manner. Xylem vessels are dead, hollow tubes. And there are three processes that we need to know. First of all is root pressure, where the roots take up ions from the soil, making the inside of the root hypertonic, and then water enters via osmosis. The pressure builds up because water has nowhere to go but up. Then we have capillary action. The rule for capillary action is the smaller the diameter of the tube, the higher the water will climb. And there are two forces at play here, cohesion and adhesion. Cohesion is the attractive forces between the water molecules, and adhesion is the attractive forces between the water and the sides of the vessel. Because water is pulling inwards, the xylem vessels here are strengthened with lignin. And last of all, we've got transpiration, where water leaves the xylem, goes from a liquid into water vapor using heat, and then the water vapor leaves the leaf through the stomata. This is also known as evaporative cooling. Next, we looked at glucose movement in plants. Glucose moves from source to sink. The source is the place of manufacture, and the sink is where it's used. Phloem is alive. It has sieve tube members. The sieve tubes are controlled by the companion cells. This is because the sieve tube members have no nucleus of their own. Glucose is made in the leaf inside the palisade mesophyll cells and is converted into sucrose. The palisade mesophyll cells have lots of chloroplasts. As I said, glucose is converted into sucrose for transport, and the transport of this sucrose is called translocation. The sieve tube members share cytoplasm, which is actively pumped by the companion cells. Then we've got another big metabolic process, cellular respiration. Cellular respiration turns chemical energy into ATP. Adenosine diphosphate is added with another phosphate to make ATP, or adenosine triphosphate. The last phosphate bond is the store of energy. Okay, first of all, we've got glycolysis that turns glucose into 2 times pyruvate. This occurs in the cytoplasm and is anaerobic and makes 2 ATP. Anaerobic means it doesn't require oxygen. But if oxygen is available, which we call aerobic respiration now, the pyruvate is shunted into the mitochondrion. The pyruvate first goes through the Krebs cycle to make carbon dioxide and 2 ATP, and it then enters the electron transport chain that makes a further 34 ATP and some water. However, if no oxygen is available and remains anaerobic, plant and animal cells behave differently. Plant cells make ethanol and carbon dioxide with no extra ATP, whilst animal cells make lactic acid, again with no extra ATP. But this prevents the pyruvate from building up and killing the cell. Next, we looked at energy flow through ecosystems, or in particular food chains. Energy enters from the sun through photosynthesis and leaves the food chain as heat due to cellular respiration. As energy passes from one trophic level to the next, only 10% is transferred, and we call this Lindemann's rule. There are 3 ways we can represent a food chain: the pyramid of numbers, the pyramid of biomass, and the pyramid of energy. The pyramid of numbers looks at the number of organisms in each trophic level. This can be a normal pyramid, or it could be inverted if there's one large producer. Next is the pyramid of biomass. Biomass is the weight of organic matter. And again, this can be inverted if there is the rapid turnover of the producer. Last of all is the pyramid of energy that cannot be inverted because of Lindemann's rule, where only 10% of the energy is passed on to the next trophic level. Okay, next of all, we've got introduced species. Introduced species are taken from another ecosystem and placed into a foreign one. The endemic species have not evolved with the same introduced species and will be outcompeted. Introduced species are successful because they have no natural predators, have a wide and varied diet, reproduce rapidly and in large numbers. They can be controlled by biological control, like the Khaleesi virus used to kill rabbits, chemical control like 10A used to kill feral cats, and physical control like the culling of feral camels. You should be aware of the parts of the microscope. The eyepiece is normally times 10. There is the body tube that connects the revolving nose piece to the eyepiece. There is the arm that connects the top to the bottom. There are coarse and fine focus knobs that adjust the level of the stage. The stage is where we put the glass slide, and the stage clips hold the glass slide in place. The light and the condenser focus light through the stage into the eyepieces. The base holds it all steady. When getting an image through a microscope, the total magnification is the eyepiece lens times the objective lens. As the magnification increases, the field of view decreases. Let's consider the image taken on the right-hand side. This was taken by Jack G and Aidan B. The field of view is 420 microns. The image diameter we're going to say is 10 centimeters, and the pink cell length is going to be 4 centimeters. So we can say that 10 centimeters equals 420 microns. We measure the pink cell with a ruler and pretend that it's 4 centimeters. So then we put it into the equation. 4 centimeters over 10 centimeters equals X number of microns over 420 microns. Then we transpose the equation so it becomes 4 times 420 divided by 10. So the length of the pink cell is calculated to be 168 microns in this case. Okay, we need to be able to work out the capture-recapture method for sampling a population. You will need to remember the equation, the number in the first sample times the number in the second sample, the number in the second sample that were previously marked. This allows scientists to estimate the size of a population without counting all of the population. So we sample a population and tag all of them. Then we resample at the same location, at the same time in the same place, say a week later. And we count how many were tagged in the second sample and use the equation. So let's think of an example. I went out into the field and caught 20 wombats and I tagged them all. A week later, I went out on the same day to the same place at the same time and took another sample. This time I caught 30 wombats, of which only 10 had tags. So I put them into the equation. 20 times 30 divided by 10, and a rough estimate of the size of the population is 60 wombats. Next is biomagnification. Some toxins, pesticides, and heavy metals are said to be non-biodegradable. Non-biodegradable means they cannot be broken down or excreted, but must be stored. And these toxins are normally stored in brain, adipose, and liver tissue. Bioaccumulation means as they are eaten, they are passed up the trophic levels. So the non-biodegradable toxin is stored each time, cannot be excreted, and must be passed on to the consumer. Biological magnification means that as the higher amounts are stored in less individuals, the concentration increases. This is why it's bad to eat apex predators. Mass extinctions. Mass extinctions have occurred regularly in the past. A mass extinction is characterized as a loss greater than 75% of species. When conditions move outside the tolerance zone of a species, without enough variation, the species will die. The Earth is currently in the 6th mass extinction, which we call anthropogenic, which is human-caused. DNA is deoxyribonucleic acid, and it's found in the nucleus of cells. DNA is composed of nucleotides, and nucleotides have a particular structure. They have a phosphate at the 5' end of the sugar. The sugar is a pentose sugar of deoxyribose, and the 3' end connects to the previous phosphate. A nitrogenous base is attached to the 1' carbon in the sugar. There are 4 types of nitrogenous base: adenine, thymine, cytosine, and guanine. And it has the structure as seen on the right-hand side. Adenine is complementary to thymine, and thymine is complementary to adenine with 2 hydrogen bonds. Cytosine is complementary to guanine, and guanine is complementary to cytosine with 3 hydrogen bonds. Now let's look at DNA replication. You need to know 4 enzymes. DNA helicase breaks the hydrogen bonds between the template strand and the coding strand. DNA primase inserts primers, and the primers give the DNA polymerase a starting place. DNA polymerase aligns the complementary bases according to Chargaff's rule, and then the DNA ligase joins the DNA backbones together. Next, we need to know about DNA biotechnology. And there are 3 types of biotechnology we need to know. First is restriction enzymes, also known as endonucleases. These are enzymes found in bacteria that cut up viral DNA, which is like the bacterial self-defense system. They cut at a recognition sequence, and the recognition sequence is normally palindromic. When they cut the DNA, it can create blunt ends, which are less useful, or sticky ends, which are more useful. Then we have PCR. PCR stands for polymerase chain reaction, and it copies sections of DNA. Step 1 is denaturation at 96, which breaks the hydrogen bonds between the template and coding strands. Step number 2 is called annealing, and this happens at 55 degrees Celsius, where we have the addition of the forward and reverse primers. Then step 3 is called elongation at 72 degrees Celsius, where the DNA polymerase from Thermus aquaticus, otherwise known as Taq polymerase, aligns the complementary nucleotides. Then last of all, we've got gel electrophoresis. Gel electrophoresis separates DNA fragments based upon size. It uses a gel chamber, a current from a battery, an electrolyte buffer, and an agar gel, which is porous. DNA is negative and is attracted to the positive electrode once the current is switched on. The shortest fragment travels the furthest, the longest fragment travels the least. The use we need to know about for biotechnology is DNA profiling. DNA profiling uses short tandem repeats, or STRs. STRs are repeating units of short DNA sequences. Everyone has 2 STRs for each location, one from their mother and one from their father. DNA profiling is also known as DNA fingerprinting, as everyone's profile is different. In a paternity suit, the baby's father is not known. The mother contributes half of her DNA to her child, and the other half must come from the father. If we look at the sample on the right-hand side, the baby has fragments at 1, 3, 4, 5, 6, 8, 10, and 11. 1 comes from the mother, 3 comes from the mother, 4 must come from the father, 5 comes from the mother, 6 can come from the mother, 8 must come from the father, so the father must be father A.